Hello everyone, myself Monu from group number 322. Today our topic is replication of DNA. One cell division, the genetic material is double and 3 occur the replication of the DNA double helix. In principle, three mechanisms of replication are possible. Conservative replication with the original DNA double helix remaining intact and a new double daughter helix being formed. Semi-conservative replication in which two daughter double helix contain each one new and one old chain. And finally, the dispersion mechanism involving the distribution of the original material among the four chain of two daughter double stranded molecules. Uh, Michelson and Stahl studied the replication of the DNA during cell division of E. coli with the aid of labeled atom and sedimentation in the shell density gradation. They produce a population of nitrogen 15 labeled cells. These cells were transferred to a medium containing N14 exclusively, in which cell division took place from the original population and from the of the first and second generation molecules. There were isolated DNA molecule and their density and radioactivity were determined. The parallel the parallel DNA The parental DNA molecules labeled with the N15 had the largest density. The DNA of the first generation was found to be half labeled and its density was equal to the arithmetic mean of the density of the N15 and N14 labeled DNAs. Finally, the DNA of the uh, granddaughter molecule was separated on the sedimentation into two zones. One zone with DNA identical with the daughter molecules and the other zone with unlabeled DNA. This may be depicted uh, schematically as follow like this. And these results are in the agreement with the semi-conservative mechanism. It has also been confirmed by other data. Template replication of DNA was also observed in vitro by Kernberg in 1916. An incubation mixture was taken that contained all four nucleoside triphosphate ATP, GTP, TTP, CTP, magnesium ion 6 into 10 की power 10 raised to power minus 3 m polymerizing enzyme isolated from cell of E. coli and negative DNA as a primer. The pH value was 7.8. In such a system, DNA is replicated. There are produced molecules identically with the primary DNA molecules. The process does not take place in the absence of one of the component or when the primer DNA is preliminarily treated with deoxyribonuclease and enzyme that destroy DNA. It was demonstrated with the aid of P32 labeled nucleotide that the synthesis of DNA is the polycondensation of monomers. When one nucleotide is incorporated into the chain, one molecule of pyrophosphate H4P2O7 is released. According to the law of mass action, the reaction is re- retarded upon addition of the excess of pyrophosphate. If its amount exceeds by 100 times the total amount of the triphosphate, the reaction rate diminished by 2 times. The newly formed The newly formed DNA has the biological properties of the parental DNA. For example, synthesis via DNA from phase phase 5 multiply 174 as the primer produce DNA which has an inactive action. Phase practical are synthesized using the polymerization enzyme DNA polymerase. One can produce synthesis polyribo polyribo polydeoxyribonucleoside capable of forming double helix in the presence of complementary uh, AT, TATA like this and like this. Uh, the free energy change during the formation of two double helix from one particle double stranded DNA and the nucleoside triphosphate may be represented in the following form like this: dash G delta G equal to 9 this, where N is the number of the nucleoside in the chain, E is the energy released when one nucleotide is incorporated into the chain. E2 is the average value of the energy elevated as a result of interaction that stabilize a Western Creek pair. The energy E2 depend on the relative content X of GC pair. This E2. The entropy change delta S is determined by the replication of the nucleoside triphosphate by pyrophosphate in solution and by the loss of rotational and translation degree of the freedom by the nucleoside incorporated into the chain. The latter factor is written in the form of K on the wall delta S, this where CPP is the conversation concentration of the pyrophosphate in solution, and this is the average concentration of the nucleoside triphosphate equal to this, and CC are the concentration of the respective nucleoside triphosphate. If there is equal, then C and TP, this the replication condition consists in the laboratory of free energy. You can see this equation. uh, the quantitative E1 and E2 are equal to about 16 and 20 kilojoule per mole respectively. The quantity Q, this equation, where Z1 and Z-1 are the partition function of for NTP in solution and for NTP bound to template by hydrogen bond, and Z2 Z-2 are the partition function for PP in the solution and for PP present in NTP and bound to template. Estimate given Q 10 raised to power minus 2. At 300K, the replication condition is fulfilled until the ratio CPP and CNTP exceeds 400. In cell and the system for synthesis, the ratio is much lower. The condition is valid if the amount of NTP is sufficient. Replication may be impeded by kinetic rather than thermodynamic factor. An analogous treatment of denaturation lead to following value of the difference between the free energy of the separate chain and the double helix. uh, Delta G, this equation where Q Q characterize the change in the intrinsic entropy of the chain per one nucleotide. One can equate Q dash to the number of the conformation of the unit of single standard chain. The unit contain 5 single bonds. The rotation about which gave rise to two or three conformation. The order of magnitude of Q dash is 10 to 100 from equation 7.51. It follows that the temperature of the helix coil transition is equal to Tm. This equation, the experimental dependence of Tm x on x, this is obtained at E80 equal to 19.7 and EGC is equal to 22.3 kilojoule per mole and Q dash is equal to 32. These crude estimates are suitable only for the assessment of the order of magnitude of H, delta H and delta S. Replication obviously requires the unwind of the double helix. Replicative system proceed with the obligate participation of DNA polymerase, which is evidently displaced along the double helix, unwinding it and synthesizing new chain. Bacteriophage alpha into 174 contain a single stranded rather than a double stranded DNA. It has been found, however, that the multiplication of this phase proceeds through a replicative step in which DNA becomes double stranded. It has been found, however, that the multiplication of this phase proceeds through a replication step in which DNA becomes double-stranded. In this case, too, it is exactly the double helix that undergo replication. This also refers to viral RNA and replication of which proceed an intermediate double helix structure. The single-stranded DNA of phase 5 into 174 is circular, just like the double helix DNA of the other phase and bacteria. In the uh, Kornberg experiment, this DNA served as a primer. However, only the circular DNA is biologically active. It exerts an infective action. Uh, Kornberg made use of another enzyme ligase for closing a new chain. The new chain were built up not with thymine but with bro- bromouracil. In order to isolate synthetic DNA, the preparation was briefly treated with DNAs so that one break in circle occurred in one half of the molecules. As a result, a mixture was obtained which contained an equal amount template circle. synthetic circle template and the synthetic open DNAs, and also double circle without break. The segmentation with the method is in the density gradient produce synthetic circle containing bromouracil. These rings provide to be capable of infective cell of E. coli. It had been stabilized earlier that the replication of even one of the 5,500 nucleotides of DNA from phase 5 into 174 led to the loss of infective action. Hence, the Kornsberg synthesis is absolutely accurate. The unwinding of the DNA double helix on the replication transcription in the synthesis of mRNA must uh, presumably be different into that involved in denaturation. The gain in free energy during the formation of new chain is responsible for a high angular momentum. The polymerase which is displaced along the double helix create local conditions that disturb the balance of interchain interaction. It is supposed to hinder the hydrophobic interaction of the bases. A new DNA is synthesized in vitro always in the 5' to 3' direction. At the same time, there are data which indicate that in vivo the two two chains are replicate, one in the 5' to 3' direction and the other in the 3 to 5' direction. Okazaki ascribed this to the discountess mechanism of the replicative synthesis. Short section of the new DNAs are synthesized on one or both chain at the replication point, uh, inversely in the 5' to 3' direction. Then these sections are joined forming new chain. If the model is correct, the last replication section of one or both daughter chain can be separate. The temporary selective inhibition of the enzyme that catalyzes the accumulation of phosphodiester linkage between the DNA chain must lead to the accumulation of new short chain. These predictions are verified by experimental which show that uh, tritium labeled thymidine is inserted into short chain which only after will until into long one. And the precision mechanism of the DNA replication in vivo cannot as yet be considered to have been stabilized. The replication in vivo starts at a definitive stage of cell growth with the participation of factors that are evidently connected with the cell membrane. Thank you.